These are embryonic stem cells. While they do not look like much under the microscope, these cells can possibly repair and regenerate damaged tissues and cells. They're pluripotent and can develop into any cell of the body. They are formed from the inner cell mass of the blastocyst and can differentiate into 3 germ lineages: mesoderm, endoderm, and ectoderm. They have the ability to treat and cure many diseases. Scientists believe they become nerve cells to restore a damaged spinal cord and pancreatic cells to produce enough insulin for diabetic. Despite these clinical and medical possibilities, embryonic stem cells have caused numerous debates. Political, social, and ethical viewpoints have clashed on whether or not it should be legal to use human embryos for scientific research. In the past year, our President Obama has lifted the ban on the use of stem cells.
I believe we have been given the capacity and will to pursue this research, and the humanity and conscience to do so responsibly.
Despite this change of law, ethical issues still remain on the use of stem cells.
It should give us hope that there's a better way forward than scientific advances that require the destruction of a human life.
The major controversy with embryonic stem cells is that in order to retrieve the eggs, you need to destroy the embryo. Opponents say that this is the same as abortion. This is the main misconception people have over stem cells. The embryos used for stem cells are from in vitro fertilization clinics that would ultimately be thrown away. Hundreds of thousands of viable embryos are thrown away every day that could be used for medical discoveries.
Oh, I get it. Well, what are the choices? What do you usually do with them?
Well, some are donated to infertile couples. There aren't very many people that need that, but that's always the first option. The rest are either thrown into these biohazard bags and will just thrown away, or they can be designated for stem cell research and used to find treatments and cures for cancer, diabetes, and Parkinson's, and more. So what should we do?
Throw them away.
But Mr. President, this is the first time in human history where scientists have had access to every single cell in the human body. These cells could save millions of lives, and we're just throwing them in the trash.
Well, I believe in the sanctity of life.
Well, that's great. So what do you think the moral thing to do is?
Well, they're human lives, and that's sacred. Throw them in the trash.
Save me now, save me now, save me now. Save me now, save me now.
The need for embryonic stem cells is large because they're the most powerful type of cell.
They have the potential to give rise to all different cell types of the body.
We're talking about whether or not we would use that group of cells to save lives.
It's time we utilize the best knowledge we have.
I think it can't hurt. I mean, when you close off that exciting an avenue of research, whatever, forget about it being a guaranteed breakthrough, a guarantee of results. It's not, nothing is a guarantee. And we never claimed that it was a surefire thing.
In the midst of creating the methods to cure certain diseases with embryonic stem cells, a new technology has emerged. Scientists are finding ways to create induced pluripotent stem cells.
A little over 2 years ago, a Japanese scientist named Shinya Yamanaka discovered that if you took a small set of genes, which are normally expressed in embryonic stem cells, and you made them expressed in skin cells, it would turn the skin cells into embryonic stem cells. And these are the so-called iPS cells, the induced pluripotent stem cells.
But these cells cannot be successful until we understand the differentiation of embryonic stem cells. We therefore have to use ESCs to determine the complex pathways of specialized cells. Embryonic stem cells will open the door on discovering the methods needed to create these induced pluripotent stem cells.
While we prolong the stem cell debate, millions continue to suffer.
And stem cell research offers hope of a cure.